So what is a glycoconjugate? First, a glycoconjugate is glucose bonded to either a lipid or protein, and it's done by phosphorylating one of the OH group and making it a better leaving group. As OH is a bad leaving group, we have to make it a better leaving group. And how do we make it a better leaving group? We make the glucose a glycosyl nucleoside diphosphate, and then it reacts with lipid or protein. So let's see how we turn the glucose in a glycosyl nucleoside diphosphate. First, we get an ATP, which then gets converted to ADP. Then the hydroxyl group, in the red hydroxyl group, changes to a phosphate group. So we have a D-glucosyl phosphate. Then in the next step, we add a UTP, uridine triphosphate, and this will help to turn the glucose into glucosyl nucleoside diphosphate. In the UTP, the two phosphates of the UTP leaves, and then the other blue phosphate gets attached to the D-glucosyl phosphate, and we get the rest of the uridine nucleoside base added here too, to the D-glucosyl phosphate. And so this is a D-glucosyl uridine 5' diphosphate. As we have D-glucosyl in the beginning, and in the end we have a uridine base, and then we have a diphosphate in the 5' of the uridine. So here we have the D-glucosyl uridine 5' diphosphate, or we could just name it as UDP glucose. Then we add the protein or the lipid, and here the UDP, which is the uridine diphosphate part, it leaves. The structure, and D-glucosyl gets bonded to the protein or to the lipid. Then we get a glycoprotein or a glycolipid.